Well, welcome back. We've been learning about the language of life that's found in nucleic acids, specifically the DNA molecule and how that gets translated into RNA. And as you can see on the screen here, that's used in making protein. I want to go in this segment, as we did in the last, into some of the details of how these processes work. How does the information in our genes, in our chromosome, how does that get converted into traits that you and I can see? Now, in this next segment, what we want to take a look at is specifically how translation occurs, now that you've been introduced to the different types of ribonucleic acids. All right, now for translation to begin, tRNA is going to bind to a start codon and signals the ribosome to assemble. And that will come in right here. And then the back of the ribosome will come, and that will cause messenger RNA to be locked into place within the ribosome. And so those reading sites will begin to be used. Notice at the top coming out of here is the new polypeptide chain that begins to be formed. Before we move on to step 2 and 3, you can see that this ribosome reading this messenger RNA molecule is not alone. There are actually several ribosomes that can simultaneously read the same mRNA molecule. And so let's take a look at the specific activity of how translation occurs. The ribosome is going to help form a polypeptide chain between the amino acids, and this is how it's done. The exposed codon that's on messenger RNA down below will then adhere to the anticodon of transfer RNA bearing this particular amino acid. In this case, it's methionine. And that's going to bring these very close to the other transfer RNA molecule as this sets in place. And then secondly, the ribosome is going to form a peptide bond between methionine, in this case, and leucine, as these two amino acids are placed side by side. And then third and finally, in this process, the now empty tRNA is going to exit the ribosome, and a complementary tRNA molecule binds to the next exposed codon here. And so once this will continue to slide along the messenger RNA molecule, forming this polypeptide chain. And once the stop codon is reached here, the ribosome will dissociate and will release the polypeptide chain so that it can fold in upon itself to form the three-dimensional protein.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
Now, I want to leave you with what I think will be a very challenging question. All along with this central dogma, we've noted that DNA is what holds the specified information needed in the production of protein. And we noted that proteins must use that information if they're going to be biologically meaningful at all. They depend upon the specific sequence of amino acids that are coded for in the process of transcription and through translation that we just looked at. But wait, isn't it also true that proteins are needed for the replication of DNA in the first place? If you recall, when we studied DNA, there are DNA polymerases, which is a whole group of proteins that are needed for the maintenance and for the replication of DNA during each life cycle in every cell, in every body of every organism that has ever lived. And so here we have a conundrum, an enigma. How do we have all the specified information in DNA in order to make the proteins when the maintenance of the DNA itself requires the very existence of the proteins that they are coded to make? And so we're left in a situation similar to the chicken and the egg down here. Which came first, the egg or the chicken? And so we can see here that at the very dawn of life, and this is important, at the very dawn of life, in the very first cells of the very first organisms in the history of life, DNA and proteins were both there, and they are both interdependent. There's no other way of understanding the story of life apart from this reality. And so the big question here is, where did the information originate that's found in DNA? And how did these first proteins arise when they're so dependent upon DNA? Well, I'll leave you to think about that. And once again, thanks for watching. Bye-bye.